Okay, we'll proceed to the next talk by Professor Ji Wu from Shanghai Jiaotong University in China. And the topic is offspring production of ovarian organoids derived from spermatogonial stem cells by defined factors with chromatin reorganization. So, Professor Wu, to you.
First of all, I would like to make many thanks to Professor Xuefeng Liu and the organizer of this meeting. And my topic is offspring production of ovarian organelle derived from spermatogonial stem cells by definite factor with chromatin reorganization. This is an overview for my topic. We know the characteristic of the germinal stem cells, including self-linear depletion and transmit genetic information to next generations. The germinal stem cells, including spermatogonial stem cells and female germinal stem cells, or FGSC. The spermatogonial stem cells identified by the Professor Rogi in 1968. They find the spermatogonial cells is from the rats and the mouse and golden hamsters. Spermatogonial stem cells and Professor Princeton development spermatogonial stem cells transplantation to the testes. Another one is the spermatogonial stem cells transplantation was performed successfully for the first time in the mouse by Professor Princeton. Spermatogonial stem cells also can keep the preservation for the male fertility. For example, can take test biopsy and isolate spermatogonial stem cells and culture in vitro. Also can make cryopreservation. And when the patient gets well and they try to get offspring, get a new baby, and they can the tissue or isolate to the spermatogonial stem cells, or spermatogonial cells can be different to the sperm, and then also can transport spermatogonial stem cells to the testes, and they get fertility and they get offspring. So also the spermatogonial cells also can generate generation of the animal model by spermatogonial stem cells. For example, with genetic transgenetic tested with transgenetics, and then isolate with the enzyme, and then the spermatogonial cells can culture in vitro, and then also can instead to injection to the sterile testes, and after 3 to 5 months, the male and the met with the female, and they get offspring with the genetic like a genetic gene and offspring for offspring. So also can modification the DNA modification for the to the spermatogonial cells to the in vitro. So also can spermatogonial cells also can in vitro develop to mature sperm or functioning sperm. Another word is in vitro production of the functioning sperm in culture neonatal mouse testes. This is the paper published to the Nature and come from the Ogawa Professor Ogawa laboratory. So for the females, it's totally different. The traditional view, they think in mammal, in most of the marine species, the production of the ovarian oocyte is thought to cease before birth. This is a literature to support this view, the traditional view. a lot of the textbook since the 1920s at the home or at the board will also describe this traditional view. However, our laboratory first time to isolate female germinal stem cells from the adult mouse ovary, and then we can make a culture FGSC, also make a cell on FGS line, and then we can transportate FGSC to the sterile mouse or ovary, and after 1.5 months, and the female met with the male, and they can get the offspring. The offspring come from the FGSC, just derived from the FGSC. This is a paper published to the Nature Cell Biology in 2009. Afterwards, another laboratory also isolate human, adult human FGSC from the adult human ovary, and they identified the FGSC. The paper published in Nature Medicine in 2012. This is so far is a research team for study of mammalian female germinal stem cells. So far we can isolate different strain from the mouse, and then make a mouse FGS line from different age and different stage also. Also, we also isolate female germinal stem cell from the rats. Also make a rats FGS line. And then for human, also we can use very small tissues or from the papier tissues, isolate FGS and make a human FGS line. This is a morphology of the human FGS line. Or another animal, we also can isolate peak FGS line, FGS, and also make a FGS line, or in the monkey FGS line, we also can make a FGS monkey FGS line. This is for monkey morphology of the monkey FGS. This is for the peak FGS morphology. We identify the FGSC is a stage between PGC and the GVO site with the single cell sequence or very small amount germ cells, female germ cell sequence. For the methylation also, methylation sequence also identify FGSC is stage of between PGC and GVO site. Also compare with the culture FGSC with the flesh FGSC, the methylation is very similar. That means culture FGSC is stable. This condition, culture condition is stable. So for the FGSC epigenetic characteristic of the epigenetic for the FGSC, we in integrative epigenetic analysis reveal unique epigenetic signature involved in the unipatency of the mouse FGSC. This is special for the FGSC characteristic. And then located of the FGSC in vivo is from in close the FGSC located close nearly surface of the cortex of the ovary. This is for day 5. This is the adult. Just the adult ovary, adult mouse ovary. We also find FGSC can differentiate to when FGS transplanted to the sterile ovary, and they can differentiate to the to an oocyte. When we FGS label with GLP, and we also can find different positive GLP positive oocyte with less FGS transportation to the ovary, also can differentiate to the oocyte. For the tracing of the characterizing the development of the transportation the FGS in vivo, and then this is the two label GFP label FGS, and then also they four we can find the FGS migrated to the surface of the cortex of the ovary. And then day 6, we can see some of the cells become large, and then day 9 and they get more large. Especially for the day 6, some of the cells get more big, and they also we can see different day 6, and then meiosis marker expression also day 9 also meiosis marker like SCP3, and the meiosis marker is SCP strand 8 expression. They indicate day 6 and day 9 the FGS goes to the meiosis, or another one is goes different to the early stage of oocyte. We was with the immunophilicity for double staining, we can see the EGFP or MVH, and double staining also can say day 2 is ovary, and then day 6 the migration to the surface, nearly surface of the cortex of ovary, and then day 6 we can see meiosis marker expression like strand A expression, and then day 9 also meiosis marker like SCP3 expression, and then day 6 we can see the around of cells with somatic the germ cells is here, and somatic cells around the germ cells. When we isolate, when we with the female transported of the FGS for female, and then met with the male, and they get offspring. The offspring we make we use the sound blood to confirm, and also with the like the offspring were identified in blood field and fluorescence to confirm the offspring really come from the GLP labeled FGS. Use the system, and we later on we try to do the fetus study of the spermatogonial stem cells. So we also make a system for the in vitro differentiation of the mouse FGS with the 3-dimensional culture model. This is how we can do with FGS with the somatic cells, and can get organoid ovary, and just like similar to nature ovary, and then we isolate to the follicle, and to after later to culture, and then we also to single cells analysis for the organoids, and then the cell type is similar to the nature or the ovary. We also culture with culture the ovary from the organoids, and then after culture, and then we can make maturation. The maturation can fertilize, and then they can development to the 2-cell and embryo development, and then we use the embryo transported to the uterus of the male mouse, and they can get offspring. We use the salt blood to confirm the offspring come from the female germinal stem cells. So this is a technology platform for our for spermatogonial stem cell study. So we think with the SSC, what happened, and we use the when we transported the female transported the spermatogonial stem cells. In other words, the SSC transported to the sterilized ovary. You can see no germ cells in the ovary, and then to what happened, the SSC can development or not. And then when we transported the spermatogonial stem cells to the ovary and then to the sterilized ovary, and then we can see because the FGS, the spermatogonial stem cells, or as I say, labeled with GLP, we can see the spermatogonial stem cells can differentiate to the oocyte labeled with GLP. You can see, and then when we take single cells with GLP single oocyte to sequence, and then we can see the Y chromosome in there. They indicate the cells from the spermatogonial stem cells because they have Y chromosome. So we also tested with the Offspring with the southern blood, also can see the Offspring come from the spermatogonial stem cells. We also take the oocyte, mature oocyte to see chromosome, and then we also can see the mature oocyte, some of oocyte including Y chromosome, some of including X chromosome. They also indicate the oocyte come from the spermatogonial stem cells. So we tracing use the technology as FGS study, and then we also tracing of the transported SSC in recepting ovary, and then you can see 2 hours, and then this is SSC, or another word is spermatogonial stem cells labeled with GLP, and this is day 2. Also, this image with confocal Mexico to tracing this cells. That day 2, we can see some cells try to migrate to the surface of the cortex of ovary, and day 3 nearly the surface. Day 4 they almost reach the surface, and then day 5 really near reach the surface. Day 6 some of cells like FGSC, some of the cells get big and get a little larger, and go different to the early stage of oocytes, and day 9 we can see more cells and more get big, and day 12 and some of can see somatic cells around the larger GLP positive cells. The 15 also can see. We also with double staining GLP and MH double staining, and to confirm the trace in the cells, like we all can see day 2, day 3, and day 4, day 5, day 6, and day 15, and we can see it's confirmed to the GLP tracing. When we try to do what happened to epigenetic or methylation, DNA methylation, what happened for the methylation? So we test H19 to we can say alternating day by day as little by little, day by day is go down to the expression actually 19, and then for the P10 is day by day is go up, and then the cross just the day 4 to 5, just similar to the just day 4, day 5, just reach to surface the day 4 reach the surface. This is very interesting. Just the H19 and the P10 is just the like a cross the 5 or day 4, day 5, and also for the implant gene or transcription factor, we also can say H19 is expression is go up, and then for the P10 is go down, and then also transcription factor special marker for the PLZF for the SSC or for spermatogonial stem cells, we can see little by little also expression go down, and then for the male germ cells marker Stella also little by little go up. This we chose the 8 gene to study, and we also make overexpression or knockdown to see what happened to cells for screening collectic implant gene and the transcription factor gene. And then we also use the organo over the organo to screen to say the epigenetic profile, and then after screening we can say I just say not what is transcriptor the germ cells from the SSC, and this profile for implant gene profile is similar to the FGSC. So we can say the morphology of the cell line. This is FGSC. This is IGSC. That means with 4 gene, we find the 4 gene H19 and PLZF and Stella, and also JFP157, the gene modified to cells, and the transgenetic or knock down the cells, and we can say the for morphology is similar, and also expression MVH, the marker of MVH is similar. Also, we also use DNA as sequence on the DNA methylation sequence, and then we can see the SSC compared with the IGS compared with the SSC is bigger different, but for FGSC is similar. Also, we also can say the related to the more close IGS, more close FGS as different SSC. For the sequence for the transcriptome, we also can say FGSC is we also can for transcription. We also can say IGSC with the same cluster to the FGSC, and then for the big difference for SSC. For the this is the example.
Excuse me, Professor, will say pop up? Oh, okay, thank you.
Sorry. Problem with the software. Okay, nearly finished. Sorry. We also try to the chromosome structure, and then after okay, during SSC conversation in IGSA or in the similar to the to IGSA, another one is transcripted to the male germ cells, and the reorganization of the chromosome structure we find. And this is the SSC, this is IGSA, and the FGSA. We can say the IGSA more close for compartment A or B, and we can say IGSA and more close to the FGSA. You can see and different assay also with the same cluster IGSA or FJ and the FGSA is same cluster and it's not a it's a different SSA. Different SSA is not a cluster, so it's IGSA more close to FGSA. So we also take an example DDPA3 expression to IGSA and FGSA, but SSA different is not SSA. So we use the organoid to say IGSA can different to the outside in vitro or not. So through the picture, we can say in vitro production of the functional outside from the IGSA. This is the 2-week and 3-week, and then we can say the IGSA can development like similar to the ovary, and this is without 4-gene transgenetic gene or knockdown gene. And then also the organelle can secrete progesterone, also X gene, and also we can see different stage of the meiosis for the germ cells. This is we isolate to follicle to culture day 0 and 2 days and 7 days, 2 days and 7 days to morphology, and then we also can see the 11 days the follicles get big. And then we will also take the follicle to outside to make a in vitro maturation. We can say mature oocytes, and then oocyte fertilization with the sperm, and can fertilize and develop to 2 cell and to develop embryo development. When we transplanted the embryo to the uterus of the female mouse, and then they can get offspring, and the offspring we also test with the salt blood and also with fluorescence to confirm the offspring from the drive from the spermatogonial stem cells. This is the rate of the maturation, and also rate of the fertilization, also number of the offspring for the how successful for this in vitro study. I try to make summarize based on the technology platform of the FGSA study. We demonstrated the successful production of the offspring from oocyte transdifferentiated from mouse SSA in vivo and in vitro. We indicated the directing of direct induction of the germinal stem cell or IGSA differentiated into functional oocyte by transduction of the H19 stellar and JFP57 and activation of the PLZF in SSA with the chromatin reorganization. This study provides a new strategy to investigate germ cell biology, epigenetic reprogramming, mammary germinal gene modification, and regenerative medicine. So make acknowledgement. I would like to make many make thanks to the Professor Lin Huo and Professor Xuejing Chen and Professor Xiaodong Zhao and so on, and PhD students and master students and so on. For the foundation, we also make thanks to the National base called the Statute Program of China and the National Nature Science Foundation of China. So thank you very much.
Thank you very much, Professor Wu, for the interesting talk. We have time for a couple of questions.
I have a question from Nanjing side. Can you guys hear me? Good. Great talk, Professor Wu. Long time I haven't hear any presentation from you. Professor Wu, can you hear us?
Oh, yeah, I can hear, yeah.
Good. I am obviously not good on biology, but I have a question about imaging. I see you have slides show of different days of development has different colors of the cell. How do you make that happen? How do you get the microscopic imaging? The basic imaging has different colors, and this is
Okay.
one. Question two is like you obviously label you do it in vitro. Still, do you worry about the fluorescent labeling or dye labeling? Does it contaminate or does it affect the biological observation or somehow affect your the study if it is in vitro? The labeling process helps you to observe it, but does that cause any trouble? Can you generally comment on that? Thank you.
Okay. Thank you for your question. Good question. For the you said for the in vitro for labeling, right? For the I just answer second question for the labeling, and we can say because the cells with just with the transgenic GLP transgenic mouse. So this means I didn't label just from isolate the spermatogonial cells, germ cells, just because the original is GLP positive cells. So we just transplanted to the positive GLP positive cells, and then with the confocal microscope, and then we can see this GLP label. And this is for immunofluorescence with the antibody, and for the immunofluorescence with the double antibody for the MVH and GLP for this part, for the right part. And second question, first question is for the different color for the to me is a different color for the offspring, right? Different color for offspring for this, right?
Such as this one, you in the top you have a few different base colors. How do you get those colors on? I mean, in each imaging you have sort of different.
For the because we use the because the mother is another original from the different because if we try to test is successful or not, must the different color because mother must be white, and we embryo just from the black color mouse, and then if we just transported to this white original white mother, and they can get different because the white color is not from the total different from the offspring, because mother is completely is not the offspring mother. It's not the embryo mother. It's just make for example just make a box, something like this. So it's different color. I don't know. It's this is you I expression I answer is Kelly or not. Sorry.
Well, yes, I think I get some clues, but we can talk in the offsite. Thank you.
Thank you. Thank you very much.
Okay, thank you very much again. Now we're gonna break for an hour for lunch or dinner for our colleagues in China, and then we're gonna meet for the last lecture block for this workshop. So
Okay,
see you all later.
Thank you. I just take outside.